Hello friends, welcome back to my tutorial channel once again. Last time we had done up to endoplasmic reticulum. Today we are going to start from Golgi bodies. So this is the next part of the cytogenetics. In plants and fungi, these are present as individual stacks. They are not interconnected and they are known as dictyosomes in plant cells. You have to remember this term, dictyosomes. It has got two faces. You can see over here. This is the concave face. This is the convex face. The convex face is known as the cis face. It is present near the nuclear envelope and it is the formative phase. There is a small transition vesicle present over here. And concave site is the trans phase. It is present near the plasma membrane. There are presence of secretory vesicles and it is known as maturation phase. Golgi body was first observed by Camillo Golgi after staining the nerve cells of barn owl with osmium tetroxide. He termed these organelles as apparato reticulare interno. In embryonic liver cells, Golgi apparatus form from the coalescence of tubular cisternae localized at the margins of rough endoplasmic reticulum. Its cis face is oriented towards rough endoplasmic reticulum from where it is formed in this way. Now, functions of the Golgi complex is it functions as principal packaging material. The materials to be packaged fuse with the cis face of Golgi with the vesicles of endoplasmic reticulum. So they remain closely associated with endoplasmic reticulum. Modification of proteins synthesized by ribosome is its chief function and that is released from the trans phase of Golgi. Golgi bodies are very important for glycosylation of proteins and glycosidation of lipids. The acrosome cap is formed from Golgi complex. It is also helpful in formation of primary lysosomes. The mucilage secretion in the root cap is also formed from Golgi. We are coming to the next organelle, that is lysosome. Lysosome is also a part of endomembrane system. This is also a single membrane bound organelle found only in animal cell. It contains about 50 times of hydrolytic enzymes. They are mainly concerned with digestion, intracellular, extracellular, all kinds of digestions are possible by lysosomes. The hydrolytic enzymes present in them are carbohydrate splitting carbohydrases, protein splitting proteases, lipid splitting lipases, and nucleic acid splitting nucleases. All enzymes in lysosomes work best at the acidic pH. That means they work best at gastric juice. It has a pH value of 4.6 to 5. I mean the enzymes of lysosomes. They have a pH value of 4.6 to 5. This low pH of the enzymes is due to the ATP dependent proton pump in them. You have to remember this. Its membrane contains special glycosylated proteins. It has got membrane stabilizers like heparin, antihistamine, corticosteroids and chloroquine. Hence, they can't be self-degraded. Now the types of lysosomes. There are basically 4 types. One is primary lysosomes or protolysosomes or virgin lysosomes. These are actually vesicles that is newly pinched off from the trans phase of Golgi bodies. Its enzymes are in inactive form. Now the secondary lysosomes or heterophagosomes. See the formation. Here the primary lysosome mixes with or fuses with the phagosomes and they together form the heterophagosome. The heterophagosome's enzymes are activated. Number 3 is tertiary lysosome or telolysosome or residual bodies. Now after formation of heterophagosome, the phagocytes engulf those particles and start digesting them, leaving behind the undigested matter. The secondary lysosome, along with their undigested matter, fuse with the plasma membrane of the cell and form. This fusion product is a secondary lysosome with secondary exocytosis. They help in exocytosis of the particles. So, they are also called residual bodies since they contain undigested matter. Number 4 is autophagosome, or these are actually suicide bags. Here, the primary lysosomes fuse with the degraded cell organelles and form autophagic vacuoles or autophagosomes. So, this is about lysosome. We are coming to the next part of endomembrane system, that is vacuoles. You can see the vacuoles over here. Just have a close look at the picture. This is a plant cell. A plant cell is having a vacuole. About 90% of the cell by volume is the vacuole. In plant cell, they are known as sap vacuole because it contains cell sap. These are single membrane bound structures. The membrane of the vacuole is known as tonoplast, and the dissolved salts present inside the sap vacuole is known as tonoplasm. Now actually there are 4 types of vacuoles. One is sap vacuole that you have already seen. It is found in eukaryotic plant cell. They contain dissolved salts. Function is to maintain osmotic pressure, and they have water soluble non-photosynthetic pigment called anthocyanin. Second is contractile vacuole, which is found in protists like amoeba and paramecium, and also in algae like Chlamydomonas. They help in osmoregulation and excretion. Third is a food vacuole found in eukaryotes and represent the heterophagosomes, as we have seen in lysosome. They help in digestion of food particles. And the fourth one is a gas vacuole. These are present in prokaryotes. Here the tonoplast is absent. In all others, the tonoplast is present, but in this case, only in this case, gas vacuole, the tonoplast is absent. These are called pseudovacuoles since tonoplast is absent. These are present in prokaryotes. They are permeable to atmospheric gases and provides buoyancy to aquatic prokaryotes. So please friends, please share this video to as many students as possible, as many students as you know, and do not forget to like and subscribe and leave your valuable comments. This is very important for me. So thanks a lot and have a best of luck.